Right, so cell division then quickly here. Okay, so what we're talking about, we've got two types. We've got mitosis, meiosis. Mitosis happens, or the way I always remember it is, it happens in your toes, or it happens in body cells or somatic cells. Okay, so that's going to take the number of chromosomes you've got. So you might have seen this number, which represents diploid. That means you've got two sets or two copies of each chromosome. That's going to produce a cell that is also diploid. Okay? Now I have seen exam questions here, sometimes in diagrams. The biological processes paper from last year had a diagram that kind of showed it like this, which represents that in mitosis, as we'll look at in a second, all the chromosomes duplicate first and then split apart. So if you see that representation there, that's representing the fact that all the chromosomes duplicate. We'll talk about that in a second. Meiosis is where we start with a diploid cell and we finish with a haploid cell. So a cell that has half the amount of genetic information. Because when we take a sperm and an egg and we fuse them together, we want to finish with a diploid cell. So we don't want too many sets of information, if that makes sense. So we'll start with mitosis. So we've got to remember IPMAT. Yeah? What does IPMAT stand for? What does the I stand for? Yeah, interphase, yeah. Then prophase, good. Prophase, metaphase, yeah, anaphase, telophase. Now, all you've got to remember are the specific sort of events that take place at each of these stages, okay? So, during interphase, everything's replicating. That's not just the DNA, that's the organelles also. And the majority of the cell cycle, if you remember about the cell cycle diagram, the point at which we're actually undergoing mitosis, which is everything after interphase, is a very short period of time. Most of the period of a cell cycle as interphase is preparing for cell division. So during prophase, after everything's divided, at rest, during prophase, all of that replicated material starts to condense. The membrane inside the cell, this is a cell here, the membrane inside the cell, the nuclear membrane starts to break down and the chromosomes start to form. So the DNA prior to, if this is inside a nucleus, the DNA prior to cell division, so it looks like that, and we call this stuff chromatin. Have you heard of that before? Yeah? Alright, so chromatin condenses then into these things, into chromosomes. Okay? That happens during prophase. And then during prophase, okay, because all the DNA is replicated, the chromosomes go from looking like this to like this. And that's something that's hard for some students to bend their head around. Okay? When we look at a chromosome individually, here's the centromere, here are the chromatids. A pair of chromosomes looks like this. So this is one chromosome, this is another chromosome. So that's a pair. But after replication, the chromosomes look like this because all of the DNA has doubled up, ready to be split. Yeah? So that's still a pair of chromosomes, it's just a pair of doubled up chromosomes. That's where that 4n number comes from that I was talking about in mitosis. Yeah? So, this is what happens in replication. So if we're looking at mitosis, during metaphase, all the chromosomes line up along the middle of the cell. So if we're talking about mitosis, the chromosomes line up above and below each other, like this, during metaphase. And then what's going to happen is they're going to be pulled apart towards opposite poles of the cell. We term this here, because it's exactly the same genetic information. This is a chromatid here, alright? These are two chromatid arms. We term that as sister, or a pair of sister chromatids. So when we talk about anaphase in mitosis, we say that sister chromatids are pulled apart towards opposite poles of the cell. The cell's a bit like the globe, right? We've got an equator and we've got poles. So the chromosomes are being pulled from the equator of the cell towards opposite poles of the cell. Alright? And it's just really being able to use that terminology. So once they're pulled to opposite poles, a new membrane forms around them. So here's one pole with one of those sister chromatids. Here's another pole with one of those sister chromatids. They pair up again, just like they were to begin with. And then they decondense. So there's another pair of chromosomes, there's another pair of chromosomes. we get a new membrane, and after that new membrane happens, this would be in telophase. If we've got two nuclei like this, all in one cell, the cell itself splits or invaginates down the middle. Do we know what we call that phase? Begins with a C. Great, yes, yeah, cytokinesis. So after cytokinesis, we've got two cells exactly the same as each other. Okay? Now in meiosis, that's slightly different. If we go back to this phase, in metaphase, when they line up down the middle, we don't need two cells that are exactly the same. So we don't need every single chromosome to give us a copy into each of the two cells that we're making. In meiosis, we're going to make cells that have half the amount of information, and also we want to have a bit of variation. So metaphase is different in meiosis. What happens is, here is one chromosome, and instead of being down here, like we've just seen in metaphase, what happens is the chromosomes line up next to each other. Okay? So that's different. This is meiosis. Now what does that enable the chromosomes to do if they're next to each other? Yeah. What's that, Alex? What is crossing over?
Oh, you just swap genes around.
Yeah. You swap genes around. Yeah, totally. Rad. So some of the information on this chromosome ends up on this chromosome and vice versa. So we create a chromosome that has different or unique genetic information. That leads to variation. So this is how like natural selection happens, yeah? Variation occurs as a result of this process. So that's the same for every single chromosome in meiosis. We're just talking about like 4 chromosomes here, 2 chromosome pairs, but that's for all 23 pairs. So instead of sister chromatids being pulled apart, like they were here, this is the sister chromatid being split apart, what happens is the whole chromosome, the whole doubled up chromosome, the pair of sister chromosomes are pulled apart. And then what happens is, when we've got 2 nuclei that have all these sister chromatids, but only one copy of each chromosome, what then happens is, these start to pull apart in a second division. So in meiosis we have 2 divisions, so they line up along the cell and then the sister chromatids are pulled apart, giving us 4 cells that all have one copy of each chromosome. So that's where each of these are haploid cells, because they only have one copy of a chromosome. They don't have 2 copies, like the original cell that we started off with. And remember, some of those chromosomes will have that little bit of change of information if crossing over has taken place. Okay? Right. That's it.